My name is Wanshi.
My name is Tay Queen. Hi, first of all, I would like to introduce our tetrapeptide 3D structures to you all. This is our tetrapeptide, which contain the glycine, cysteine, alanine, and tyrosine. On the left is our N-terminal, is from glycine, and on the right we have the C-terminal, is from tyrosine. As you all know, tetrapeptide is contain 4 amino acids, so we have 3 of peptide bonds. The first peptide bond is located between the glycine and also the cysteine, and the second peptide bond is located on here, which is between the alanine and also the cysteine, and the third peptide bond which is located at here, and it connected the alanine to the tyrosine. Okay, the disulfide bridge likely to happen at the sulfide molecule, which the sulfide molecule will undergo the oxidation reaction to losing 1 electron. Therefore, the sulfate ion have the affinity to bonding with another sulfate ion to form a disulfide bridge. Furthermore, the hydrogen bond will take place at the nitrogen molecule and also the oxygen molecule when they are surrounded by the delocalized electron. So this is our first structures, which N-terminal is start from glycine and the C-terminal is from tyrosine. This is our second structure, which N-terminal is from tyrosine and C-terminal is from glycine. The difference between structure 1 and structure 2 is their N-terminal and C-terminal located at different amino acids, so it gives different function and also chemical property to the tetrapeptide.
Hello, my name is Zilan and I am in charge for the second question. As you can see, we have already got this one is ribose and this one is deoxyribose. Both of these structure have 2 types of different structure. The first one is 3-endo and the second one is 2-endo. As the name implies, 2-endo means that the carbon number 2 of the ring structure has its position above the plane of other carbons in the rings. The same applies to the 3-endo, but the third carbon is above the plane. Same applies for the deoxyribose. The second endo is second carbons move upwards the plane, and the 3-endo is third carbons move upward the planes. Now what I have in my hands is uracil. By combining the uracil and ribose, we has made the nucleosides. This one, they can connect it to ribose sugar, but not to deoxyribose. Also, to bind this, the N1 of uracil need to bind with the C1 of ribose. The way they bind is this OH will go up and this one is N1. The H will broken and form water, and the nitrogen of N1 will meet here, will bind here. That's all. And then from this form, to form the complete nucleotide, we need to have a phosphate. With this phosphate, we can bind one of the oxygen with the 5 carbons of ribose, and then we have created the nucleotides called uridine monophosphate.
Hi, I'm Wan Shen. So I will be explain about question 3. So this is the 3D structure about the alpha D glucose. So this one is the first carbon, second, third, fourth, fifth, and the sixth carbon 6. So how do we determine the alpha or beta glucose? Is based on the alpha OH group. So if the OH group at the carbon 2 is face upward, so it's alpha. So this one is the beta D fructose 3D structure. So beta D fructose is one of the cyclic forms of fructose. So it consists of 5-member ring structure. So it's 4 carbon with 1 oxygen atoms. So this one is the beta D glucose 3D structure. So this one is the C1, C2, C3, C4, C5, and C6. So how to determine this one is the beta D glucose is based on this one, C2. The OH group is at the bottom of the C2. So second question is the maltose is formed by 2 glucose molecules. So this one is the first alpha D glucose. This one is the second alpha D glucose. So they bind with this is the 1, 2, 3, 4. This one is the C1. So C1 will bind with the C4 carbon and form alpha 1,4 glycosidic bond to form a maltose. So for now, I will explain why maltose is a reducing sugar. So just now, like I said, maltose is formed by 2 alpha D glucose, right? So after, if one of the glucose is taken away, so here it will form aldehyde group, which is C110. So this has a free anomeric carbon, and which means the aldehyde group, and the aldehyde group will allow the maltose to act as like reducing agent in redox reaction. As you can see, this is a conjugate from carbohydrate and also protein named acetylglucosamine. Acetylglucosamine play important role in our body health. Dysregulations of acetylglucosamine cycling has been implicated in various diseases such as cancer, diabetes, cardiovascular disease, and etc. So for the next, acetylglucosamine also innate the immunity by detecting the bacterial peptidoglycan. So the formation of mannose-6-phosphate is targeting on the lysosomal enzyme oligosaccharides.
Acetylglucosamine can be found widely in nature since it is the monomer of polymer called chitin. Chitin is a polymer that creates the exoskeleton of insects and other creatures. This acetylglucosamine can also be found in membrane of cell. When it is hydrolyzed, it can release this. When the chitin is hydrolyzed, it can release this acetylglucosamine, and when this acetylglucosamine is put into a strong acid, it can change the acetyl group into amide. And this acetylglucosamine can also responsible for blood group specificity.
That's all from us. Thank you.